You see a salamander, a small, slick amphibian, skittering under a wet leaf. It's not much to look at. But if a predator bites off its leg, something incredible happens. The salamander doesn't just heal. It doesn't form a stump, it grows the leg back. A perfect leg, complete with bones, muscles, nerves, and skin. It grows back the toes. It grows back the ankle. It's a flawless copy of what was lost. And it's not just legs. A salamander can regenerate its tail, its jaw, parts of its heart, and even sections of its brain. It's a biological superpower. It makes our own healing look primitive. When we lose a limb, it's gone forever. We get a scar, a permanent reminder of the damage. Our bodies seal the wound. That's the priority. Stop the bleeding, prevent infection, and move on. The salamander's body has a different priority. It aims for perfection. It wants to be whole again. So the question hangs in the air, a question scientists have been asking for a very long time. Could we ever do that? Could a human ever regrow a lost arm or a leg? The idea sounds like science fiction, but the science behind it is very real, and researchers are starting to peel back the layers of the salamander's secret. They're trying to figure out if that same magic is locked away somewhere inside our own DNA. To understand the challenge, you have to first understand the salamander's process. It's a beautiful and complex dance of biology. When a salamander loses a limb, the first thing that happens is pretty similar to us. The wound is sealed. A layer of skin cells moves across the surface. This is called the wound epithelium. It's a protective cap, but this is where our paths diverge. In a human, underneath that cap, a flurry of activity begins. Cells, called fibroblasts, rush in. They start spitting out collagen, a tough, fibrous protein. This is the stuff of scars. It's like a quick patch job. It's strong, it's fast, but it's not functional tissue. It's just filler. Our immune system helps this process. White blood cells called macrophages swarm the area. Their main job is to clean up debris and fight bacteria, but they also send out strong signals that promote inflammation and scarring. It's an emergency response. The body is screaming, close the breach now. In the salamander, something much more subtle happens under that wound cap. The cells at the site of the injury do something amazing. They dedifferentiate. That's a key word. It means they go backward in time. A muscle cell forgets it's a muscle cell. A bone cell forgets it's a bone cell. A skin cell forgets it's a skin cell. They shed their specialized identities and become like generic jack-of-all-trades stem cells. They form a clump of these amazing versatile cells right at the tip of the stump. This clump is called the blastema. The blastema is the engine of regeneration. It's a small mound of potential. It contains all the instructions needed to build a new limb. The salamander's immune system plays a very different role here. Its macrophages are different. They are anti-inflammatory. Instead of shouting signals to scar, they send quiet signals that encourage the formation of the blastema. They help clean up the site without triggering a panic response. They create a calm, controlled environment for rebuilding. Then, the blastema starts to grow. The cells divide, and as they divide, they start to redifferentiate. They get their old jobs back, but in a highly organized way. They listen to signals from the body. These signals tell them where to go and what to become. It's called patterning. The cells know to form a bone in the middle. They know to wrap muscle around it. They know to weave nerves and blood vessels through the muscle. They know to create a hand or a foot at the very end. They even know where to stop. A salamander that loses half a leg doesn't grow a leg and a half. It grows exactly what was missing, and then it stops. The level of control is breathtaking. Nerves are critical to this whole process. If you cut the main nerve leading to a salamander's limb, it won't regenerate. The nerves don't just carry signals for movement and feeling. In the salamander, they release special growth factors into the blastema. These factors are like a constant stream of encouragement, telling the cells to keep dividing and building. Without that encouragement, the process stalls. So we have the blastema, a calm immune system and helpful nerves. This is the salamander's toolkit. Now let's look back at us. Why don't we have it? The most obvious answer is scarring. Our bodies are built for speed, not for elegance. For most of human history, a deep wound was a death sentence. Infection was the real enemy. So our bodies evolved a rapid response system. Scarring is that system. It's better to have a sealed off stump than a gaping wound that gets infected and kills you. Regeneration is a slow, energy-intensive process. It takes weeks or months. In the wild, that's a long time to be vulnerable. Scarring is a survival strategy. It's a trade-off. We traded regeneration for a better chance of surviving the initial injury. Our immune system is another major hurdle. It's aggressive. When we have a major injury, our immune response is a storm of inflammation. That inflammation is good for killing germs, but it's terrible for delicate rebuilding. The pro-inflammatory signals essentially tell our cells to lock down and form a scar, not to experiment with becoming stem cells. The environment at a human wound site is chaotic and hostile. The environment at a salamander wound site is calm and constructive. And our cells are stubborn. A human skin cell is a skin cell. A muscle cell is a muscle cell. They are locked into their jobs. This is called terminal differentiation. It's a feature, not a bug, of our biology. It provides stability. It prevents cells from suddenly changing their identity, which can be a hallmark of cancer. We have very powerful genetic locks in place to keep our cells in line. The salamander seems to have a key to unlock these cells temporarily, use their potential, and then lock them back into place safely. We've lost that key, or at least, we've hidden it very well. There's also the genetic component. The salamander genome is bizarre. It's enormous, 10 times larger than the human genome. For a long time, scientists thought most of it was junk DNA, but now we know that a lot of this non-coding DNA is actually regulatory. It's like a massive switchboard that controls which genes get turned on and off. The salamander's genetic switchboard seems to have a big, glowing button labeled regenerate. Ours doesn't, or if it does, it's covered by a thick layer of dust and a do not press sign. So the barriers are significant. Scarring, an aggressive immune system, stubborn cells, and a different genetic operating system. Well, it sounds hopeless, but it's not, because science is not about accepting limits. It's about testing them, and researchers around the world are tackling this problem from fascinating angles. One approach is to learn from other animals. It's not just salamanders. Regeneration exists on a spectrum. A planarian flatworm is the undisputed king. You can chop it into hundreds of pieces, and each piece will regrow into a complete worm. A starfish can regrow an arm, a zebrafish can regrow its fins, and even parts of its heart. We can't do that, but we're not at zero either. Children can regrow fingertips. If a child under the age of about 7 or 8 slices off the very tip of their finger above the nail bed, and the wound is just cleaned and covered, it will often grow back perfectly, with the fingerprint and nail. This ability fades as we age. It suggests we have some latent regenerative capacity, but it gets switched off. Why? No one knows for sure, but it's a tantalizing clue. There's also a special kind of mouse, the MRL mouse or healer mouse. These mice were discovered by accident in a lab. Researchers noticed they were closing up ear punches, a common way to identify lab animals without a scar. Further studies showed they could regenerate damaged heart tissue and cartilage much better than other mice. They're not salamanders, they can't regrow a whole limb, but they heal better than their peers. Scientists have been studying their genes, trying to find what makes them different. It gives us a mammalian model to study, which is much closer to us than an amphibian. These examples show that the genes for regeneration might not be entirely foreign to us. They might just be dormant, suppressed by other pathways that favor scarring. The question then becomes, can we reawaken them? Can we convince a human wound to act less like a human wound and more like a salamander's? This is where the work gets really exciting. A team of scientists at Tufts University, led by Michael Levin, has made some incredible progress. They work with African clawed frogs. These frogs, like us, don't naturally regenerate limbs. When they lose a leg as a tadpole, they grow a thin, useless spike of cartilage. It's a scar. Levin's team decided to try and kickstart the regenerative process. They designed a small device they called a biodome. It's a tiny wearable bioreactor made of silicone filled with a special gel. This gel contained a cocktail of 5 drugs. Each drug had a specific purpose. One was to reduce inflammation. Others were designed to encourage the growth of nerves, blood vessels, and muscle. They were trying to replicate the chemical environment of a regenerating amphibian limb. They would amputate a frog's leg and then seal the stump inside the biodome for just 24 hours. Just one day of treatment. Then they would take the device off and watch. What happened next was astonishing. The frogs didn't grow the useless cartilage spike. Instead, over the next 18 months, they regrew a leg. It wasn't a perfect leg. It didn't have all the right toes, but it was a real functional leg. It had bones, muscles, and nerves. The frogs could use it to swim and push off surfaces. A 24-hour treatment had triggered a months-long cascade of regeneration. This was a landmark experiment. It showed that the barrier to regeneration isn't necessarily a lack of information in the cells. The frog cells knew how to build a leg. They just needed the right trigger. They needed a little push to get them started on the right path, away from scarring and toward rebuilding. The biodome provided that push. It created a little bubble of a pro-regenerative environment, similar to a blastema, and that was enough. Now, a frog is not a human, but this is a proof of concept. It suggests that we might not need to rewrite our entire genome to be more like a salamander. We might just need to find the right cocktail of drugs or growth factors to apply at the right time. We might need to trick our own wound sites into behaving differently. Another major area of research is, of course, stem cells. Stem cells are the body's raw materials. They are unspecialized cells that can develop into many different cell types. We have them in our bodies, mostly in our bone, marrow, and fat. The idea is that we could harvest these cells, grow them in a lab, and then inject them into a wound site to help with healing. But it's not that simple. Just injecting a bunch of stem cells into a stump won't create a new arm. You would likely just get a disorganized lump of tissue, what's known as a teratoma. The real challenge is organization, or patterning. How do you tell the stem cells to form a bone here, a muscle there, and a nerve in just the right place? This is where scaffolds come in. Scientists are building biodegradable 3D scaffolds, often in the shape of the missing part, like a piece of a bone. These scaffolds can be seeded with a patient's own stem cells. The scaffold provides the structure, the basic blueprint. It guides the cells, telling them where to grow. As the cells build new tissue, the scaffold slowly dissolves, leaving only the natural, living replacement. This technology is already being used to regrow small sections of bone or cartilage. But scaling it up to an entire limb is a task of immense complexity. An arm has hundreds of muscles, dozens of bones, and miles of nerves and blood vessels, all arranged in a precise architecture. Building a scaffold for that is a huge engineering challenge. Then there is the frontier of gene editing. Tools like CRISPR give us the ability to directly edit our DNA. In theory, we could use this to turn on the genes that promote regeneration and turn off the genes that promote scarring. Remember the MRL healer mice? Scientists have identified some of the genes responsible for their enhanced healing. A gene called Lin28A, for example, is active in young animals and seems to play a role in regeneration. Reactivating this gene in adult mice improved their ability to heal. Could we one day create a genetic therapy that activates a whole suite of regenerative genes in human cells? Perhaps, but this is a risky road. The genes that control cell growth and division are a delicate balancing act. The process of telling a cell to forget its identity, divide rapidly, and then form new tissue sounds an awful lot like cancer. And this is the dark side of regeneration, the biggest hurdle of all cancer. Cancer is essentially uncontrolled cell growth. Regenerating animals like salamanders have a fantastic ability to grow new tissues, but they are also incredibly resistant to cancer. They have somehow figured out how to unleash this powerful cell growth and then stop it on a dime. They have powerful tumor suppressor genes and mechanisms to kill off any cells that go rogue. We don't fully understand how they do it. If we were to start switching on powerful growth promoting genes in our own cells without having the salamander's robust cancer suppression systems in place, the results could be catastrophic. We could be trading a lost limb for a deadly tumor. This is perhaps the single most important safety concern that scientists in this field are working on. We need to understand not just how to turn regeneration on, but how to keep it under tight, reliable control. So, where does that leave us? Will you or I ever see a day when a person can walk into a hospital and have a lost arm regrown? Probably not. The timescale for this kind of breakthrough is likely measured in many decades or even centuries. The complexity of the problem is just too great. Think about what it would take. You don't just need to grow a limb, you need to connect it. The new nerves have to find their way up into the spinal cord and brain and make the right connection so the person can control the arm and feel with it. The new arteries and veins have to perfectly link up with the existing circulatory system. The immune system has to accept the new tissue, and the whole process has to be perfectly patterned. The arm has to be the right length. The hand has to have 5 fingers, not 6 or 4. The thumb has to be opposable. The amount of information required to build a limb is staggering. In an embryo, this information unfolds through a precise, genetically programmed dance. The salamander's blastema seems to be able to tap back into that embryonic program. We don't yet know how to do that. We don't even fully understand the program itself. But this doesn't mean the research is a failure. Far from it. The quest for limb regeneration is forcing us to learn more about our own bodies than ever before. The knowledge we gain along the way could lead to incredible new therapies that fall short of regrowing an entire arm, but would still change medicine forever. Imagine being able to heal a damaged heart after a heart attack, not with scar tissue, but with new, beating heart muscle. Imagine being able to regenerate the cartilage in an arthritic knee, ending chronic pain for millions. Imagine helping spinal cord injury patients regrow damaged nerves, restoring feeling and movement. Imagine helping burn victims regenerate healthy skin instead of getting disfiguring scars. These are the more realistic, near-term goals of regeneration research. They are not about creating superhumans. They are about enhancing our own natural healing processes. They are about convincing our bodies to fix themselves better. The work on the frog's leg, the study of the healer mouse, the advances in stem cells and gene editing, all of these are building blocks for that future. We are learning how to dial down the scarring response. We are learning how to talk to our cells in their own chemical language, nudging them toward repair instead of a patch job. We are mapping the genetic circuits that control healing. Each discovery is a small step, but the steps are adding up. The salamander remains the ultimate inspiration. It shows us what's possible in biology. It proves that a vertebrate animal, an animal with a spine like us, can possess the blueprint for perfect healing. That blueprint might be buried deep within our own genetic code, a relic from a distant evolutionary past. We may never be able to access it in the same effortless way a salamander does. Our biology has taken a different path. But we have something the salamander does, and we have conscious inquiry, we have science. We can study the salamander, learn its secrets, and then try to translate them into our own language of medicine and technology. We can't differentiate our cells at will, but we can build bioreactors and formulate drug cocktails. We can't naturally form a blastoma, but we can build scaffolds and seed them with stem cells. We are trying to use our minds to achieve what the salamander does by instinct. So, could we ever regenerate limbs like a salamander? The honest answer is maybe, but not anytime soon, and it probably won't look like the natural process we see in that little amphibian. It will be a medical procedure, a combination of drugs, bioengineering, and cellular therapy. It will be a hard one, human-made version of regeneration, but the journey to get there is already transforming how we think about healing. We are moving away from the idea that damage is permanent. We are starting to see the body as a system that can be guided, persuaded, and help to repair itself in ways we once thought impossible. The salamander is our teacher, and we are slowly, carefully learning its lessons. The ultimate goal isn't to become a salamander. It's to become a better, more complete version of ourselves.